It's my pleasure to introduce Otto from Oxford, who will be talking about networks and single-cell data, multiscale methods for selecting gene expression signals.
Hi, so yeah, I'm Otto, and I'm a joint PhD student at Ludwig Cancer Research and Mathematical Institute at Oxford. And I'm going to be talking about some joint work between all these people here. And yeah, so it's another not necessarily neuroscience talk, but I'll be talking about things, networks and signals, and so hopefully there's some relation to what you look at. Okay, so what is the kind of problem that we've been looking at? So often data comes to you as this graph on network, right? As I'm sure you're aware, and you often have a collection of signals on this network, right? So these could be certain features that inherently come from the network. So if your network was originally a set of points and then you made this into a network, you have the original coordinates of these points and Rn. And often these features are actually interesting biologically, for example, right? So for me, these features are going to be genes, and we want to try and understand this network in terms of these genes themselves, right? So we will have this very large collection of these features. I mean, there may be like 20,000 graph signals. So we want to try and find only the ones which are very relevant to our network. And I'm particularly interested in the single-cell transcriptomics data. So what is transcriptomics? This is the kind of what's called the dogma of molecular biology, right? Where you start with this DNA in your cell, and all of our cells have the same DNA apart from a few exceptions, but all our cells are different, right? A T cell in your body is very different to epithelial cells, etc. So there's something else happening, and that is what parts of the DNA get turned into different proteins, right? And this goes through this process of transcription onto RNA, which then eventually gets turned into proteins. So if we can understand this RNA, then we have a very good window into what the cell is doing, right? And then this leads us on to things like trying to understand gene regulation or just the identity of cells themselves. And so this collection of RNA transcripts inside a cell is called the transcriptome. And biologists are generally interested in, okay, I have these two different types of cells. What are the genes that drive the differences between these cells? Okay, which genes are highly expressed and which are lowly expressed between these different cells? So kind of the usual way to do this is that suppose I have these two populations of cells. I just kind of aggregate their expressions for one particular gene. So how much each of that gene is being turned into RNA for each of these two cells. And I can do some statistical tests between these two populations. Okay, so I have these red and blue curves being two populations of cells, and they represent the expression of this particular gene. And I can do this t-test, and if these expression patterns are very different, this gene is definitely different between these two populations. And this has sort of worked well in the past with past technology, where we have this data called bulk data, right? Where for each sample, you sort of smush all the cells in together in this sample, right? You can imagine it like having a smoothie. You have strawberries, bananas, and grapes, but then you just whiz them up, and there's no real way to understand the different constituents of the RNA from different cells. But in the last decade, we've had this massive revolution in the kind of data we can get for biology, which is this single cell data. And this allows us to look at these single cells and sequence them. And this is magnified some of the problems, though, right? So now we have tens of thousands of these cells and 20,000 of these genes. How on earth do we sort of analyze this data? So the usual method is to do something like this, right? So you start with some collection of data points. If each of these data points are cell profiles, and you create some network out of these cell profiles by looking at their global transcriptomic profiles, right? So if globally the gene expressions are similar, you join them together in some network, right? And this gives you some network here in the middle. But biologists like to understand things in terms of cell types, and this is at the moment just some network. So then what you do is you usually cluster it, or use community detection, as you've heard about today, and that gives you, this set of communities. And then from this, you can go and do what I described earlier of this, just using these statistical tests. And for each gene, then you have some p-value, which tells you how significant it is. And then this allows you to rank the genes based on interestingness, right? So if there's a very low p-value, it means it's interesting. But this process is problematic, right? Especially this last bit. I mean, when you say something has a certain p-value, it kind of gives you the feeling that it has a certain statistical weight. But there's a sort of chicken and egg problem where you've made this network from these global transcriptomic profiles. You've made these communities based on these global profiles. And then you do this sort of hypothesis test where you assume that they were independent to some degree. So, I mean, what that's really saying to us is that these p-values are only exploratory, right? We shouldn't give them necessarily any kind of strong weight. They're just a way to rank them by how interesting the genes are. And I mean, then again, what even is a cell type, right? We're making these community structures from this network. In many cases, cells are very different, right? And it's kind of easy to tell the difference between them. But there are definitely some processes in biology where cutting up into discrete types, it doesn't really make any sense. So for example, I mean, I like T cells, and they're very plastic. They kind of change between states quite easily. And there's, I think, a lot of headache of trying to decide, okay, well, this is one new subtype of T cells, this is another subtype, when really they're just kind of states in some more continuous notion of a T cell. And then, things like cancer, especially, is very different from all the different neighboring cell types. And obviously, if you have something that's a cell type which is changing from one stem cell to a more developed cell, there's no way to cut this and say, okay, something definitive has happened here necessarily. So maybe let's not look at the community structure of the networks, right? Just treat them as continuous objects. And can we still do this sort of ranking of our genes based on these networks? So here's another example about, again, okay, we're using these networks, and we do this community detection. These statistical tests are not very good. They rely on these communities we've defined, right? So in this very simple example, right, I just have these, imagine that each of these represents a cell, and I have two large blobs and sort of bridging cell in between. And I kind of cut them vertically, right? So I go across, and this is my decision of the two communities. And each of these vertical cuts, I do this statistical test for this gene, and I find that, okay, well, sometimes it's significant, this particular gene. So, sorry, here, red means it's highly expressed gene, black means it's lowly expressed. But this varies wildly, right? And okay, maybe it's not so bad on this example, but if you have 20,000 of these genes, I mean, the ranking is going to be very different as you move across, right? Okay, so how can maybe we fix this? So we need some way of finding these interesting genes, which I agree is entirely subjective, but we want to do this in a way which isn't necessarily in the arbitrary clustering. And so here, when I talk about the networks that I'm using, these are cell-cell networks, right? So each point is a cell, and these nodes are linked if these global profiles are similar enough. So, okay, one way of defining what interesting means for some graph signal network is to define what it means for it not to be interesting. And I mean, you can kind of, maybe one way of saying that is if this signal is probably likely due to noise, right? Its value wildly goes up and down as you move from node to node in your network. So for example, the top one, I mean, if you think the red ones are highly expressed, the black ones are lowly expressed, then there doesn't seem to be any correlation of this gene expression with the underlying network. But if you look at the bottom one, okay, hang on a second. Yeah, the right blob is mostly red, the left blob is mostly black. So maybe that's interesting. But we have a network, right? We know what each cell connects to. So we can just look at each of these edges and just measure the difference of this gene expression value across each of these edges, okay? And then square them. I know this cut is destroying my lovely equation. But okay, we have some measure then. We just measure the difference of this value across each of the edges. And I mean, probably most of you know what the graph Laplacian is. You probably, oh yeah, sorry.
Sorry, you may have mentioned this. So you talk about edges, but here you have the vertices. Are we to assume vertices that are close to each other probably have an edge?
Yeah. It's not actually my picture, but yeah, yeah. So I mean, imagine that, yeah, that we have edges here.
Okay.
So it turns out this notion of testing the differences across edges is related to this thing we've been talking about a lot, this graph Laplacian. And this graph Laplacian is, okay, you can make a matrix out of a network, right? If I have this matrix here, this network here, I can look at its degree matrix, which is just this square matrix where all the edges and rows are indexed by the nodes of the graph, and they have, the diagonal is the degree of that node, right? So node number 1 here has degree 2, so we have a 2 here. We also have an adjacency matrix, which is just recording whether there's an edge between node I and node J. So here between 1 and 2, we have a 1 because there is a node between them. And if we take the difference between these matrices, we have something called the Laplacian matrix. Why this is nice to look at is that, if we think we have one of these graph signals, right? So this expression of a certain gene, if we hit it with this matrix, thinking of this graph signal as a vector, then what it measures is the difference between, as I said, the value on a node compared to the average of its neighbors, right? And then if we hit it again with our vector, we get exactly this differences along the edges that I talked about. So if we really want to measure this, it's also useful to think about where it's going to come from, this Laplacian. But measuring the difference along the edges is a bit problematic in the sense that if I scale my graph signal, my expression of a gene, then this will change how much is changing along each edge. So I want to do some normalization. So I divide by this, the norm of the signal, right, this FTF. And this whole quotient is called the Rayleigh quotient. And it's measured, so if it has a lower score, it means that this signal is much less randomly varying. So, we like to think of this as more interesting. And, if you can also do much more of these different normalizations, there's a whole sort of literature on which is the best. But just think about it as this quotient between the sum of the differences of the edges divided by the norm. Okay, so, this has been around a while, but it's also recently was applied on some of this single cell data. So this is these infiltrating lymphocytes into tumor. And I think this is just a TSNE plot of this whole genome expression data. Along the x-axis is this rank of this Rayleigh quotient. So this is the Rayleigh quotient. And if you look at the genes which have a high rank, so near 1, so a low score, then you can see that they are reasonably consistent across your network. Okay, so you have to imagine we have all these edges going on here. If you look at something which has, a very low rank, i.e. a very high quotient, then, I don't know if you can really see this, but it's very noisy, right? It doesn't really relate to the global picture of your cells. Okay, so this seems to be doing what we want, but there's a bit more of a problem. So it ranked, just to go back, it ranked this CD8 gene as more interesting than this FGFBP2. But which is more interesting, right? If you think the left one is more interesting, raise your hand. Or the right one, or both. Yeah. Sorry to those who are here on Tuesday. I think both interesting for different reasons. One is clearly dividing your network into 2. One is very localized to one particular arm of your network. So we should have methods which maybe not give us a linear ranking, but a multi-linear ranking. Okay, so there's three problems with this method, and we came up with three different methods to deal with it, right? So the first one, what was nice about doing this community detection is that you're splitting your graph into different bits. So when you do the statistical test, you know where the difference is happening. It's happening between cluster A and cluster B. So we sort of lost that by just looking at the global differences along of these gene expression values. And as I just showed in the last slide, this expression can happen on different scales. And it'd be nice if there's a way to look at one particular scale. What are the genes that are happening very locally within our cell-cell graph versus larger scales? And then finally, you might also, especially in biology, have some other data with that you've observed, right? So if you're looking at how an embryo is developed over several days, you may have sampled this over several days, and it would be useful if you could use that information to also separate out your genes, right? So if we could have, each of these requires a different method, but I'm going to go through them. So our first hero is Eigenscores, okay? So what was nice about the Laplacian is that it contains all this other interesting data, right? And a lot of this will probably be familiar if you think about dynamics on these brain networks. Because of particular properties, it has very nice eigenvalues and eigenvectors, right? And these also have a very nice interpretation in terms of your graph. So if you look at the eigenvectors corresponding to low eigenvalues, these correspond to bits of your network which are very distinct but still connected, but they're almost separated, right? And then as you continue to go up these higher and higher eigenfunctions, you get these higher and higher harmonics of your graph, right? So you can kind of think of these as splitting your network apart, but in only a kind of a rough sense, right? So imagine I have this map of France and I look at I think this is the fifth eigenvector, then it's sort of splitting it into three different parts if you look at the positive and the negative parts of this eigenvector. And then, so on and so on. So you kind of have this splitting of your network into communities without actually having to make communities. So if we get this Rayleigh quotient we were looking at before, and we can kind of do this Fourier transform using these eigenvectors, and then only look at the components along each of these different, I suppose, modes of the graph, then we kind of have how interesting this feature is, but spread out about each direction of the graph. So here's just a very simple example. So I have this original network like this, where I have four major components. This is the first eigenvector, splitting it into two. This is the second eigenvector. And I have some collection of these graph signals, right? So red here is high, blue is low. And you can see they have kind of a continuous shift as we go along, separating out these signals depending on which part of the graph they're highly expressed in. And if you had some signal which is just not very interesting, because you're normalizing by the norm, this ends up just falling in the middle. So if you look at this periphery, you end up finding kind of the signals that are interesting for that particular component. So I'm gonna not really talk about this that much, but you can, as I said, do this ranking for each of these eigenvectors, and you get a new ranking of your interesting genes, and they increase in frequency depending on how much resolution you want to look at. Or it's quite nice is this visualization of your genes. Okay. So I can just cut the first few of these eigenvectors after I do this transform and produce some visualization. So here, each of these nodes represents a gene, so a graph signal. And this is using some UMAP on this cutoff of the first 17 eigenscores. And you can see we have, David likes to think this looks like a turkey, I'm not sure. But each of the sort of branches of this turkey corresponds roughly to one of the larger communities of this original network, which is here. But what's nice about it is that it's still maintaining some of this continuity. Okay, so if I go over here, you can see that, okay, this arm represents the genes which are highly expressed on this particular subset. I don't know if you can really see it. They're monocytes. But what is nice is that because it's continuous, is that as I go around the periphery, I have a gradual change in the expression across the monocyte population, right? So if I think something is interesting, I still have a sort of context of what it's related to in terms of other genes.
So
Yeah.
what's that original data set?
So this is blood cells, PBMC data. So it's quite nicely clustered because, I mean, if you look kind of on a broad scale, you have lots of, I mean, the B cell and the T cell are broadly very different. Yeah, so this is why we have such a nice clustering. But then if you want to kind of zoom into monocytes, or you have all these different subtypes, yeah. Okay, so our second hero is this multiscale Laplacian score. So this is relating to the problem, as I said, of if you have a gene expression which happens at different scales. And we heard earlier today about random walks, and of course, Laplacians are related to random walks. So a random walk is where you start on one particular node and then with a certain probability jump to neighboring nodes, depending on the weight of your edges around that particular node. Okay. And random walks are nice with Laplacians because they both relate to this idea of diffusing across a network. But the Laplacian itself that I talked about only really describes one type of random walk, where I just hop once to one node and then I hop again and I hop again. And the problem is that this is kind of one type of scale that I'm looking at, right? I've decided my edge is one scale. So it would be nice if I kind of sort of adapt this random walk to deal with a different time frame of how fast I want to move across the network. So, I mean, I'm not going too much in the details, but what happens is that I can simply reframe this as a continuous problem. So I have some parameter t and solve it. And what I end up with is this new random walk Laplacian, but it has a parameter which tells you, okay, what is the probability if I start at one random node, or start at a node and I wait to a certain time t, and this t could be any positive real number. So I can take that and plug it into this equation I talked about earlier. And what it boils down to is that I have the same Rayleigh quotient or Laplacian score, but now I have some continuous time parameter or scale parameter that I can look at, right? So, okay, why is this useful? Why do I want to have this scale parameter? Okay, imagine I have, this very simple network, right? Where I have 3 different community scales. So this very small community of just, 4 nodes, and a medium community and a large community. And I have 2 different gene expressions or graph signals or features, whatever you like to call them. And one is very consistent on this low community, but it's not really consistent on everywhere else. Likewise, the other one is very consistent on this larger community, but it's not consistent anywhere else. But if I look at this random walk, then what I can notice is that if I start on this small community and I only wait a short period of time, I'm still going to be in this small community. So I'm still going to think of myself as consistent with all my neighbors. But if I change to a larger time point, well, then suddenly I start spreading through the other communities and I suddenly realize I'm not a very consistent signal. But if I look at the large community, on quite a much longer time period, I'm still very consistent because I have so many more neighbors to visit. So if I look at these 2 different scales and 2 different time periods, I can sort of separate out genes which are expressed at different scales, okay? So I'm plotting some time point 1 versus the difference between this score on 2 different time points. And what you see is that this signal here, it falls all the way down here, but the signal on this very small network is now not very consistent when I start thinking this larger time. So on real data, so this is the same infiltrating lymphocyte data, although we've now used a slightly different projection, so it looks slightly different. And we see the same thing, right? So here I have this one gene IFI27, and it's only expressed on this very small population of cells, right? And very consistently. On the other hand, NKG7 is expressed on a very large collection. I think these are all NK cells. And it's also expressed on a number of other cell types throughout the graph. But I've now been able to separate them out and say, okay, well, if you're interested in things that are very local, look at this portion of genes. If you're interested in very broad genes, look at this other portion. Okay, and maybe just as a quick aside, because this is related to diffusion, there are actually kind of useful ways to detect which time points to look at by looking at your community. There's something called variation of information, which tells you how stable communities are. So if you kind of look at these, if you want to boil down the time points to look at. Okay. Okay, finally, we have the persistent Rayleigh quotient. So we have some extra time data that comes along, and we want to say, okay, well, which genes go upwards with this time versus which genes only go, up one pathway across this time data, right? So you might have a gene which is generally related to, okay, a cell going through this development phase versus a gene which is actually driving one cell to become some other cell type. So, okay, well, how can we handle this data with the time data? Well, one thing we can do is chop our graph up into pieces depending on the different time steps. So initially you might think, okay, well, I start at the whole graph, right? So by the way, this graph here, I'm only really thinking of some sort of tree where we start at one cell state and we sort of divide into different cell states later. So you might say, okay, well, let's start at the initial time and then continually increase our tree as we go forward in time. But it's not very interesting to look at from this graph perspective because we only have one connected component or community, and we don't really see much changes, right? It's only one connected component that always continues for each of these time steps. But if we go the other way and look at, okay, we start at the very end time point, so we just look at the time points D and E, and then slowly look backwards in time, then we see, okay, well, D and E did have some common ancestor cell type. And then, they were eventually joined by B and C, and we have a much more interesting detectable changes if we look backwards in time along our network. But the problem remains that, okay, well, what do we do then, right, once we've cut our network into all these different time steps? Well, I mean, we could create this new Laplacian feature of these time steps, but this doesn't really tell us how each time step relates to the next one, right? It might tell us that at one time step we have these two connected components, but it doesn't tell us, okay, at what point do these connected components join, and how does this relate to some gene expression or graph signal? So it turns out there is a nice way to do this coming from something called network circuit theory. And the way this works is that I choose some subpart of my network that I'm interested in, and I delete everything else, right? But I do this in a way that preserves a lot of the geometry of my network. And so you can see here, right? So I'm interested in keeping these red nodes, and I want to get rid of the blue nodes. I can delete them, but as I delete them, I keep the information in the red nodes about how easy it is to go to the other red nodes while going through these blue nodes, right? And this whole process is called the Kron reduction of this network. Just for those who are interested. The way this works is that you reorganize your Laplacian based on the nodes you're interested in. So you shift them all up to the top left, and then you do some Gaussian elimination to get rid of the nodes you're not interested in. And it turns out this is all very well defined, and this gives you some nice Laplacian. But okay, so we go back to our very simple example of some cell type C that at some point later splits into two cell types A and B. Okay. Well, we're looking backwards in time, so we start at just thinking about the points A and B, and we do this Kron reduction, so to get rid of the point C, and we now introduce introduce a half-weighted edge between A and B. And what this does is tells us that at some point in the past, there was this cell type that split in between them. And then using this information is useful because all the things about the Laplacian, etc., and the Rayleigh quotient rely on having connectivity to tell the difference of gene expressions. So if we were to just get rid of C, then these are two disconnected components, and there's no way to ever measure the difference of a gene along them. So we can do this iteratively. So we have some whole long period of time points. We say, okay, we're not interested in anything before time t1, and we then look at some other point t2, and then we kind of reduce t1 to t2. But the reason why I'm doing this whole thing in the first place is to try and separate out signals that have some sort of different relationship to time. Okay, so if I look at, again, this very simple example of the C splitting into A and B, and imagine the 4 different types of signals I can have on this graph. Okay, so I can either have that this gene is highly expressed at point C and say point A, but not expressed at point B. So this might be interesting as telling us that this gene is helping the cell to drive along down one particular lineage, versus something where gene is only expressed at both A and B, but not at C. And this could, for example, be a gene that only exists after the cell has decided its fate, right? So it'd be nice if we could separate these out. And then by looking at these different time points, this is exactly what happens. And so if you look above the line, you find these genes which are only expressed down one lineage versus other genes which are expressed down only in the children, not the parents. So we apply this to some real data, and this is an example of some developing mouse cells where we have these liver cells, stem cells, hepatoblasts that develop over several days into cholangiocytes and hepatocytes. So you can sort of see this is the network we start with, and we have time going this direction and our filtration going the other way. So we look at all the genes. Each of these dots represents one gene or graph signal. And again, we do this comparison of just looking at the whole graph itself versus reducing it just to the last few days and trying to look backwards along, to see the changes of that gene expression before. And we see the same phenomenon where these gene expressions that relate to one particular lineage, say TUB5, do appear above the line, right, as we would expect them to. And gene expression signals which only present in the daughter cells appear below the line. Sorry, these guys, ALDOP and MT2. Okay, so that was the 3 methods for these gene expression signals. Yeah, I hope that was interesting for you guys. If you want to see our paper, it's up there. And yeah.
Great, thank you very much. Questions for Otto?
Yeah, just a quick question about the data set in the final slide that you got. These are different cells, right, across time? Not the same cell.
Yeah, they're different cells. So you've sampled, they're different batch experiments as well.
Yeah, so do you, how do you align the development time across these cells? Do you know there's a defined starting reference time? You know what I'm talking about? Because different cells have their own clock time, right? Which is different from your real physical time.
Yes. So, just first of all, this is some public data. It wasn't my experiment, but I think what they did is that you start at development day 0, and most of your cells are there's some variation along them. But then as you sample across different days, you see the spread in the population as they move through this development period. So you kind of agglomerate all of that, and then you hope that the ones that have a similar clock appear close together. But you already, does that answer your question?
Yeah, no, I was just curious how the method works for when you're uncertain about the reference time of these. But I think your answer is they do start at the clock time and the cell time is basically a line from.
Yeah, I mean, this is on one particular, in this experiment, it's very nice that you have some developing embryo cells and you can go back multiple days. But there are also many experiments where, you can only really estimate the development time. So, you have these pseudo time methods. But I mean, fundamentally, the sort of method would still be the same. You just have to be slightly less sure about this, exactly about the pseudo time. But yeah.
I guess you can still find this clusters, you just can't color them really the same way.
Yeah, exactly. I mean, I've seen the pseudo time can be well done. It can also be very badly done. I've seen some awful uses of it. But so as long as you're reasonably confident about your pseudo time, then it should hopefully work the same way.
So I guess your filtration doesn't need to be restricted on the network being a tree. Because you need to use an example that doesn't seem to be a tree, for example. That's an actual network.
Yeah, I mean, in fact, it doesn't. I mean, I only think about it in terms of trees to sort of motivate, particularly for this problem, right? I mean, you could use this. I mean, you could do the same thing for any filtration you like. You wouldn't necessarily see this sort of split where these certain expression signals come above the line and below. And it's also, I mean, it's slightly unclear to me why exactly this still happens, right? Because the Laplacian is very combinatorially dependent on the underlying graph. So, I mean, if you look at these eigenvectors, you can convince yourself that for this very simple case, this will always happen, that you split the two different types of signals up and down below. But if you look at this, I mean, there's no, I feel like there's quite a bit of a leap in what I'm saying. I mean, for such a complicated network, you could have this combinatorial adding of this edge could vastly change what's going on. So this is kind of something I'm interested in looking into.
I was just curious about some basic properties of this Kron reduction. So we can have an intuition about it. Is there something, what is maintained when you eliminate the nodes? Something like weighted path distance from each node on the one community to the other, or?
Yeah, so what is preserved is the effective resistance between the nodes. So this is why it comes from the network circuit theories, where you think of each edge as having a certain resistance given by your weight. And why they wanted to do it is that they wanted a simpler representation of the network, but they could still trust that the electrical properties are the same. But effective resistance also has some very nice properties. I mean, you can turn it into a distance if you just take 1 over the resistance. Or no, that is a distance. But yeah. It also has, I mean, related to TDA, there's also some nice properties. I mean, it preserves like the persistent Betting number. There's a whole, they come from like something called persistent Laplacians as well.
Any other questions? Great, let's thank Otto again.